Okay, the cell cycle, mitosis, all parts of vital cellular processes that need to happen for us to survive. We know mitosis is asexual reproduction for growth, for repair. We progress from G1 to S, G2, and then to mitosis, prophase, metaphase, anaphase, telophase. So that's the big picture, right? That's the whole process. But something we got to kind of think about is there are going to be like little checkpoints. There's going to be little points, think of this as a pathway. We're going to have little inhibitory points that uh, make sure that we're okay before we continue onwards. Yeah. So if you're going to go on vacation, right, you're going to start traveling, well, you got to kind of check to see, do you have enough fuel to get you to where you need to go, right? You want to be prepared. That's kind of what the cell does. So there's going to be certain qualifications that we have to meet before we can go to the next level, something like that. Before you can take anatomy 1, you got to have all the content of biology 1306. It's a prerequisite. So there's certain prerequisites that we have to meet before we can proceed to the next part of this cell cycle. So when we talk about checkpoints, think of them kind of like that, right? Like little barriers. So before we can progress from G1 into S, we have to stop, we have to analyze, we have to be sure that all the proper qualifications are in place, and then we can continue. So there's going to be 3 critical checkpoints, and this is good for the exam. I would know this for your exam. There's 3 critical checkpoints that regulate the cell cycle, and they're going to be the G1 checkpoint, G2 checkpoint, and M checkpoint. So gap 1 checkpoint at the end of gap 1, gap 2 checkpoint at the end of gap 2, and the M or metaphase checkpoint in metaphase. So all of these checkpoints have to work properly. They have to do their job. They have to be strict. They have to regulate and not allow the cell to proceed to another part of the cell cycle if it's not ready to do so. So I'll let you kind of guess here for a moment. What primary aspect is the G1 checkpoint looking for? Now, what's the whole point of G1? So you should know the whole point of G1 is to make ATP, to undergo cellular respiration, to generate all that ATP. It's going to give us enough fuel to make it through the S phase. So G1 checkpoint is checking for that. It's checking to see, is the gas tank full? Is the cell phone battery fully charged? Is our ATP supply ample enough to carry us all the way through S phase? We don't want to run out of energy halfway through our DNA replication process. Right? So G1, checking for basically, is there enough ATP? So yes, there is. Okay, that barrier open. The barrier opens, we can pass through. So we replicate our DNA. We go to G2 and we have another checkpoint, my G2 checkpoint. 1, did we replicate our DNA properly? Was that DNA replicated the way it's supposed to? 2, do we have enough ATP to go into mitosis? So there's a little bit more regulation that happens in G2 than we had in G1. So if we have some errors with the DNA, we can't send that damaged mutated DNA to another cell. So that cell will not be allowed to continue onwards. We could maybe send in our DNA ligase and some of the replication enzymes to go redo the replication possibly. If the DNA was perfect, great, but we still need enough ATP to progress to the M checkpoint. So these are, again, important phases of our cell cycle, the cell cycle control, the cell cycle checkpoints. The M checkpoint, if you remember mitosis metaphase, we have to line all those chromosomes in the middle. We have to wait for all the chromosomes to get to that spot. So this is the role of the M checkpoint. I don't know if you traveled to field trips when you were a kid in elementary school, but if we went to the museum, we couldn't go back home. We couldn't go back to school until all the kids made it back to the bus. Right? So we gotta wait. We gotta wait. Little Johnny's not here yet. We gotta wait. Little Susie's not here. We gotta wait. So we can't proceed onwards until everybody is back to the bus. We can't proceed until all those centromeres, all those chromosomes are on that metaphase plate, that midline of that cell. Once they all get there, then we're clear to go. We can separate an anaphase and proceed onwards. So normally we have no issue, we have no problems with the cell cycle because of these 3 checkpoints. They do their job properly, and normally that's not a problem. Again, these checkpoints are like stop signs. They halt our progress. They don't let us go to phases that we're not ready for. So again, normally they do their job. We do run into some situations where the cell checkpoints are not functioning properly. Right? If we analyze, let's say, the cell cycle. So let's say we analyze injuries that we've had. I don't know if any of y'all have had some bad lacerations, cuts, ugly cuts that needed a lot of stitches. Well, those cuts hurt. They may bleed, whatever, but eventually they heal. They heal with scar tissue, but they heal. I don't know if you have any interesting broken bone stories, right? The bone breaks. Well, that bone hurts. That broken bone hurts a lot, but that broken bone heals by the process of mitosis. So that's the purpose of mitosis, right? For growth and repair. So we have to cycle around and around and around and around and around. The more times we go to mitosis, the more repair that we allow to happen. So ugly cuts heal, broken bones heal. I don't know if you've ever analyzed nerve tissue. Right? This picture, most of you probably don't recognize this character. This guy, Christopher Reeves. Christopher Reeves was the Superman when I was a kid. Right? You watch Superman now, it's that Cavill guy, another younger actor. Back in the day, this guy was Superman, Christopher Reeves. And he was riding a horse, had a weird accident. He fell kind of strange. When he landed, he broke a cervical vertebrae that severed his spinal cord. Right? So he became, from one day to the next, one day he was the actor that played Superman, very good in shape. Next day he was a quadriplegic, paralyzed from the neck down. So he had a terrible spinal cord injury. Yeah? The moral of the story, right? So he damaged the spinal cord, and people that that have had injury to the spinal cord, or let's say maybe elderly grandparents that are having issues with Alzheimer's, or sort of a deterioration of brain cells, of neurons, of nerve tissue. So what happens when we damage nerve cells, right? Those nerve cells are not able to repair themselves. So if they're not able to repair themselves, that tells us that they're not going around their cell cycles. So when we go back to our checkpoints here, nerve cells, when we're a child, when we're infants, and early in our life, damaged nerve cells can regenerate, to some extent. Once we reach adulthood, we're out of luck. If you damage a nerve cell, that's it. Those nerve cells get stuck right here at gap 1, and they stay there. Right? So right now, if you damage nerve cells and you live to be 100, those nerve cells are going to stay damaged all the years from now till when you reach 100. They're never going to repair themselves because they get stuck here. They get stuck. They're overly regulated. The cell cycle regulation is so strict, it does not allow them to proceed to mitosis where they can repair. Right? So that's a problem. It's an overregulation. Now, do you think that there are certain scientists, labs, researchers that are trying to find that checkpoint. They're trying to understand the checkpoint and figure out how to sort of knock that barrier down to allow nerve cells to go repair. How would the world be different if we can figure out how to allow nerve cells to go repair themselves? It would be a different world, right? It would be a very, very different type of environment there. So interesting. Maybe with the Bridges research program, maybe some of you can get into some of these labs. Maybe you can be part of that team that figures that out. And how would the world be different if nerve cells can regenerate, right? What would happen to any paralysis? What would happen to cerebral palsy? What would happen to Parkinson's, Alzheimer's? These things would be illnesses of the past if we can just figure out how to make adult nerve cells function like regular cells, yeah, like skin or like bone. What would happen to our memory? We would not forget things because those cells would stay fresh and be able to regenerate. And it would just change the world. So that would be amazing if that could happen within our lifetime. Yeah. So that's one problem of overregulation. Now we come down here. I know you've heard the term cancer. Well, what is cancer? Cancer is an underregulation. Cancer allows cells to progress from phase to phase to phase to phase to phase when they're not supposed to. Right? So in adult nerve cells, the parent is being too strict. No, you're grounded till you're 100, right? Over here, the parents are not doing nothing. There's no regulation. The kid's doing whatever they want. They're at Walmart, little pain in the ass kid is just messing stuff up at Walmart. That's kind of like cancer cells. They're in no regulation. The cell cycle has lost regulatory control over them. So cancer cells are going around and around and around and around, making abnormal, irregular cells that are not supposed to be formed. So it's quite amazing. Two very opposite problems that are basically having a real huge impact on human health, right? So in our lifetime, you know people that are going to have Alzheimer's. In our lifetime, you're going to know people that have cancer, right? So both of these illnesses are due to cell cycle issues, right? Problems with the cell cycle. If we can make cancer cells act like nerve cells, make nerve cells act like cancer cells, how different of a world would we live in? Right? So I wish I had the answer. I wish I did. I would have a lot more money in my bank account if I knew how to solve these cell cycle issues, how to stop cancer, how to allow nerve cells to regenerate. So again, we need a lot of your bright, young, enthusiastic minds to go into these fields and really, really make an impact. Again, I know a lot of you want to go into nursing, but how many lives, how many millions, billions of people, in the future can you help if we understand how to just let nerves do what they're supposed to do, heal, make cancer cells stop dividing. Right? So again, something to consider, and that's my motivation to you all to really look into these research careers, these biomedical research careers. They're going to open a lot of doors for you, and they're going to provide you an ability to have a huge impact on human life, right? And make a huge contribution with your careers for sure. So with that, let me stop this one, and then I'm going to formally address cancer in our next discussion.